We'll be moving on to our next presenter. Chen is the IPM advisor for Sonoma, Mendocino, and Lake County. Good morning, Chris. If you want to share your screen, today he will be talking about a different kind of pairing, grapevine rootstocks and site conditions.
Thanks, Cindy. Thanks for having me all. Just quick correction, I'm the viticulture advisor. Cindy is our IPM advisor for this region. No, you're good.
I read it too quickly. Thank you.
Yeah, I know it's a long morning. But yeah, thanks for having me. I'm going to talk today about pairing your rootstock for your vineyard with your site location with consideration to pests and abiotic stressors. I've added abiotic stressors in there because my research program focuses pretty heavily on climate adaptive viticulture. And both of these topics relate to that main umbrella. Just brief introduction. I probably know most of you, but I'm Chris Chen. I got my doctorate at UC Davis. I've been the viticulture advisor in the North Coast region of California, Sonoma, Mendocino, Lake counties for almost 3 years now. So let's get going. And if you have any questions, please put them in the chat. I'm happy to stick around and answer them at the end. So I'm going to start off with a little history lesson with rootstock use and why we use them. So rootstocks have been around for a long time in viticulture now. They're considered an integral part of growing grapes in pretty much every grape growing region of the world. But originally, they were introduced to combat grape phylloxera in the late 19th century by French viticulturists because phylloxera made its way over there and it was wiping out vineyards. And these were developed using wild grapevines that were native to the Americas. The wild grapes that they used, they evolved alongside grape phylloxera, and they can tolerate, most of them can tolerate their feeding damage on the root systems, which Vitis vinifera could not. So for many decades after the introduction, the majority of rootstock species were developed from crosses of what I call the big three species. These are Vitis riparia, Vitis rupestris, and Vitis berlandieri. Each of these were selected for very specific traits that were desirable for vineyards, along with their natural tolerance to grape phylloxera, and two of them, their ability to root and graft. So this is just a little map of what the spread of phylloxera looked like pretty quickly. It was in the United States, was, I believe, the place it was brought from, went to Europe, started wiping out vineyards. So we still have this problem in most viticultural regions today, so we still use rootstocks. And today they're used for all sorts of purposes. Pest tolerance is kind of the universal benefit of rootstocks since grape phylloxera exists in almost every viticultural region around the world. Sometimes nematode resistance is needed as well, so there are grapevine rootstocks for nematode resistance, tolerance or resistance. Another common benefit is their tolerance to abiotic stressors like drought, or the opposite, anoxic inundation. If you're dry farming, if you have bad quality soils or heavy lime in the soils, stuff like that. So matching the rootstock to the site condition is very important, and it can help with erosion or managing vine vigor. It can help suppress pathogen symptoms, for instance, grape family virus with O3916, but also in some cases, grapevine scions, which are Vitis vinifera, can also do this. A great example is the Thompson seedless grapevine, which can be riddled with grapevine trunk diseases or fungi and still produce pretty acceptable fruit. And then finally, rootstocks can modify the site conditions through soil impacts, or they can alter the fruit size through vine-water relations. Most commonly, rootstocks exist to help scions adapt to specific site conditions or common vineyard stressors. They're also selected by nurseries for the ease of their propagation, their grafting, and their rooting, as well as their popularity. It's also not the norm nor required to slap the rootstock name on a wine label, so we can use kind of whatever we want and not have to, send that information out with the wine. But that's not the case with scion cultivars. And sometimes consumers purchase wine by the cultivar they're familiar with, especially if it's a single varietal wine. So we actually have a really big benefit in not having to put the rootstock on the bottle because we can use whatever best suits our situation. And these are the most common rootstocks in California. And likely for most of you, this is not a huge surprise that these are the most common. 1103 Paulson, I like to call it the universal rootstock because if you have a need for a rootstock and you don't know what to plant, a lot of folks will go right to 1103P. Freedom, Ramsey, 3309C, and SO4 are the other most common ones in California. And their crosses are here too. So this is kind of what it looks like in a pie chart as far as the popularity and the amount of planting that was done with these rootstocks in 2022. Their popularity holds true across most of California. With some cultivars, they're a little disproportionately planted in specific areas, like Freedom in the San Joaquin Valley. We don't have a lot of Freedom rootstock in the North Coast part of California, but it is very ubiquitous to the San Joaquin Valley. Other rootstocks like 1103P are planted universally due to their really wide range of suitability at different sites. And this data is not easy to come by, right? This doesn't show up in the acreage report. So we have to actually collect this data if we want to know what the most popular rootstocks are in our state. And you can see 1103P makes up a pretty large portion. Because of the strong skew toward a handful of rootstocks, nurseries often produce more of the most demanded cultivars. That could limit us in what characteristics and tolerances that we introduce to our vineyards via the rootstocks. For instance, like 1103P, if that's not the best choice for delaying bloom or avoiding spring frost, but if that's all that's available for a grower looking to plant a vineyard, then the grower will likely have to install frost protection infrastructure to deal with that. Just an example, right? That's not always true. But if another rootstock is less available or unavailable that could solve the problem, it becomes an issue of whether or not that choice is available at their nursery, right? So there are hundreds of rootstock varieties, each with their own traits that might be beneficial in certain circumstances. And I think we should be using more of them in more situations. My advisor during my graduate school program, Andy Walker, used to say, don't try to pick the right rootstock because there are many right rootstocks. Just avoid picking the wrong one because the wrong one can really mess you up. Just a brief note about scion selection. Scions are in a very similar situation as rootstocks, right? There are a few that dominate the plantings in California. These two situations, the rootstock and the scion bottlenecks, they are one of the best avenues to potentially addressing climate change in vineyards through diversification of varieties, right? So around 40% of wine grapes in California are either Cabernet Sauvignon or Chardonnay, with the others being a mixture of a lot of different varieties. And this makes sense, right? For growers that want to sell the grapes or for wineries that want to sell bottles because that's what consumers buy. But as we're facing what I'm going to talk about later with changing climates and how it's affecting pests and how it's affecting abiotic stress in vineyards, we'll probably want to explore the wide range of genetic adaptation that's available in the many other scions that exist out there, and rootstocks for that matter. The best way to do that and still get by, I think, is to test different cultivars to see what performs best in a changing hyper-localized climate. And then that can help you decide what to plant in the future and maybe even find a market for it before you plant it. So back to rootstocks and specifically their response to pests. Because almost all commercial rootstocks were introduced for their resistance to grape phylloxera, that is the majority benefit we get from rootstocks for pest tolerance. There are also rootstocks that are resistant to nematodes and other pathogens. And this is just kind of like how rootstocks were developed. But, quick note, some of these resistances were by accident, so that's great. But there are a few rootstocks that were developed specifically to deal with an individual pest or disease, like 03916 with nematode tolerance and grapevine fan leaf virus, or the GRN series of rootstocks, which are great for nematode resistance. Most pest tolerance in vineyards comes from the rootstock, and most of the rootstocks get their pest tolerance from some trait found in a parental, often wild grapevine. For example, Muscadinia rotundifolia is one of the rare sources of ring nematode resistance in grapevines, and it's been used to develop many rootstocks, some more successful than others. It also seems to be difficult to root, which makes it more difficult to work with. There are some scions with pest tolerance too, but this requires that the scion is not 100% Vitis vinifera, and that can be a problem for some of the wine grape industry, definitely. It also can change the characteristics of the fruit. So we look at rootstocks for pest tolerance most often. Just wanted to take a break from rambling and kind of specify the difference between tolerance and resistance as far as it relates to plants. So tolerance is the most common mechanism of damage mitigation in plants, and it refers to the plant's capacity to withstand damage, usually feeding damage, and that's very common. Resistance is a term that's used a lot, but is a less common mechanism in plants and in grapevines. It's also doesn't mean that the damage isn't impactful to the vine. It just means that you are resisting it. Usually this is done through like limits on pest reproduction or feeding efficiency. A really good example of this is white mustard. White mustard, if you plant it in a monocover, so there are no other plants with it, it helps resist nematode pressure due to the glucosinate it produces in its roots. Nematodes don't like that compound, so their populations tend to go down in the fields that are only white mustard, which is hard to do, right? So what are the limitations of pest tolerance and resistance? Most resistance or tolerance of pests occur in the rootstock, with some exceptions, right? Like the PD, Pierce's disease, and powdery mildew resistant scions, which exist and haven't really been taken up yet by growers, but may become necessary. Soil-borne pests are the most targeted for development of these tolerant traits in rootstocks because the rootstock is the intermediary between the scion and the soil, right? So it's the one that deals with the soil pests. Rootstocks and scions are unable to eliminate pest problems almost always. I'm sure there's a case or two out there where that might have happened, but that is not what we look for when we're looking at pest tolerance or resistance. They're mostly able to just tolerate the damage or limit the damage of the pest through limiting population growth or feeding efficiency. So the pests will be there in the rhizosphere, in the root zone, regardless of what tolerant or resistant rootstock you plant. It's just how impactful it is to your growing operation. And there are rootstocks that are, like I said before, developed specifically for tolerance of specific pests. The GRNs are the grapevines resistant to nematodes, which is a great name, I think. Very straightforward. These are relatively new rootstocks, right? They've been around for a while, but they haven't been widely planted. So they were developed to tolerate nematode feeding, and not just like dagger nematode or root knot nematode, but a lot of different nematodes. And you can see they are the ones that were released were GRNs 1 through 5, and they have different crosses of different parents, some wild, most of them wild actually, and some cultivated too. And just looking at how they perform, right, GRN 1 through 5, I'm going to start with GRN 5 because it is the most difficult to work with from what I've understood. So GRN 5 is less popular than the other ones. And GRN 1 is really good. It has pretty moderate vigor and rootability or graftability, and it's resistant to most nematodes that are problems in vineyards. Tolerant, I would say. GRN 2 and 3 tend to have, from what I've seen in data, tend to have higher vigor in the scion they're grafted to. They root fairly well, they graft fairly well, but they have limited tolerance to other nematodes like ring, citrus, and lesion. GRN 4, kind of in the middle there. GRN 4 is moderate vigor. It's decently rooted and grafted. Again, moderate tolerance to the, I would call them secondary problematic nematodes here. But GRN 1 is kind of the most unique one and the one that is gaining a lot of popularity. 2 and 3 are also very popular. But GRN 1 has a specific parent, Muscadinia rotundifolia, and has taken up the trait of ring nematode resistance from Muscadinia rotundifolia. Both parents have issues. Both parents of this grapevine have issues in the vineyard, right? I don't know why I put this. That's not true. Vitis rupestris is fine, but Muscadinia rotundifolia is known to be hard to root and graft in nurseries. So GRN 1 does have this ring nematode resistance. It has a pretty good success rate from grafting, which is great considering one of its parents is Muscadinia rotundifolia, and it does have decent vigor. This is the one that most of the people I work with, most of my clientele prefer to plant if they're going to choose a GRN. So getting away from just rootstocks, I wanted to take a look at how pests and diseases are responding to observed climate change in the area, the vineyards. So the changes that we see are likely the result of direct and indirect impacts of changes in temperature, increases in temperature, specifically increases in winter temperatures, which are very important for pest and disease survival, changes in developmental timing of predators and parasitoids, changes in distribution and range of host plants as well. And I have a fun map for that later. Because of these and more impacts of climate change, it's pretty likely we're going to see changes in the ranges, not the migratory behavior, the ranges of pests and diseases, and the preferred locations of where they grow. Their overall overwintering success, the interactions that these pests and diseases have with species in the vineyard, including grapevines, and then the effectiveness of pests, predators, and parasitoids or beneficials. Insects in particular have several ways to respond to climate change based on these different metrics of impacts that we were talking about in the last slide. These are the three most commonly cited, right? So moving to a climate that's more suitable for them. If it gets too cold or too hot or too wet, and they're able to move somewhere else, they will establish populations there. They might shift their phenology to correspond with the local changes in the environmental conditions, like changing the time that they hatch, for instance, time of the year. Or they could adapt to the new conditions and associated impacts on the ecosystem in other ways. This is kind of a map of a specific pest and how it moves, how it's expected to move, right? Due to increased temperatures mostly. So increasing temperatures will alter the climate of a given region, no matter where you are. And vineyard pests will move to their preferred climate regions if they're capable of doing that. Often they do that on our vehicles or on our boots or something like that. For California and most of the northern hemisphere, they'll probably be moving north and toward coastlines. So you can see that the blue line here in this figure represents a hypothetical limit to this pest range. As temperatures increase, that limit increases north or goes north, and they can spread a little further. But this is a map from the conservation corridor, and this shows what it would look like for a number of species in the next 100 years or so, what their ranges would be moving to. And what surprised me about this specific video or figure is that even coast redwood trees are expected to be moving north. So in California, we're known for our coast redwood trees, and they do exist in like Oregon, in some other places, but they're expected to move their range north in the next 100 years. So this is one of my favorite maps to show people, especially when I'm talking about this topic. And pathogen success also is bookended more or less by temperature extremes. So my favorite example is fungal tolerance to high temperatures. So there's a lot of fungi that cannot exist in certain areas because it is too hot there during summer and they can't survive that. Or it's too dry, they can't survive that. That is changing because those regions are changing, right? But it's not just because the climate is changing, it's also because the organism itself might be changing. They need to adapt to hotter climates, or they need to open more niches for themselves in different areas. And for the former there is a human fungal pathogen example, Candida auris, which has always been a pathogen of humans, but had a hard time surviving in the specific temperature and environment of our bodies. And so it would typically only infect humans with immune disorders. Recently, by recently, I mean in the last 10 years, it's been infecting humans that don't have immune disorders because it's been shown that it has evolved or adapted to the temperature of the human body now. And that's been attributed to adaptation to higher average temperatures in their ambient environment. So that's a problem. And we haven't seen that in agricultural systems yet, but I have a feeling that it might occur at some point. Viral temperature adaptation could be similar too. Viruses they adapt rapidly to new conditions because they have so many generations in such a short time span. So we need to look out for that too. So I'm going to swap over for a little bit to abiotic stress and rootstock selection. That was kind of like how biotic stress affects our choice of rootstocks and what we should be looking out for. but we're going to look at abiotic stress because abiotic stress is just as important in my opinion. So the information in the text here comes from a study that was published in 2022, actually. No, sorry, 2021. And it looked at the change in key developmental stages of grapevines from spring to fall. And this was in Central Europe, but it should hold true for most places that are experiencing similar situations, and as far as ambient temperature increases. But between 1985 and 2018, every major phenological event in a vineyard occurred at least a week earlier than in 1985. So this is something we expect to see, and this could affect a lot of aspects of vineyard management, as well as grapevine development and berry phenological development as well. And in Santa Rosa, where I have an office, Sonoma County, I made this graph in 2022, but I just want to show you the difference in growing degree days or heat accumulation hours in Sonoma County of California, right? A major grape growing region. So 2012 is the orange line and 2020 is the purple line. And those are the ones I really want to highlight in this example here. If you look, if we're looking at a fixed growing degree days, in this case, I don't know why I chose 1800, but it's 1800. You can see that in 2020, we actually got to 1800 growing degree days in Santa Rosa, about 50 to 60 days sooner in the year than in 2012, which means that the grapevines likely were developing faster than in 2012. And this can also affect your insect pests as well. But by the end of the season, we also had more heat accumulation than in 2012, and by quite a bit too, by almost 1,000 growing degree days, which moves us into a different climate classification, a different Winkler index, which means that the pest and disease and abiotic stress problems that we had in 2012 are going to be different than we have in 2020 in this example. And this is something we need to look out for year after year and not just rely on, well, we always do this practice at this time of year. Sometimes that might be too late. It might be too early for a year that doesn't quite hold up to historical records. So as far as abiotic stress goes in vineyards, there are, a lot of them, but the main ones are heat, which affects the berry shrivel and phenolic degradation, as well as developmental rate of the grapevines and pests. Frosts and freezes, which affect your green tissues and can split your trunk, as well as overwintering survival for pests. Solar radiation or just sunburn, I guess, that can increase phenolic degradation. Water, as far as drought and inundation of water go, depending on the time of year. Drought can affect your yields. It can affect senescence of the leaves, early senescence. It can decrease photosynthesis. Inundation can change your root abiotic environment, root biotic environment so that it is abiotic, which means your grapes aren't getting enough oxygen. And that can lead to senescence. There's also chemical drift and physical damage, which are human imposed most of the time. This can, really just physically damage the vines. So how does climate change affect abiotic stress? This is, I think, pretty straightforward, right? We know ambient temperatures are increasing steadily. This is the most impactful as far as winter temperatures go, right? Because we have less overwinter curing of diseases like Pierce's disease, and we have better pest survival in winter as well. And if it gets too warm in winter, we will not achieve the quiescence required for grapevine buds to break. That would be very problematic for a grape growing region. Overall heat accumulation hours are increasing, right? I just went through that in that figure. Precipitation patterns are less predictable than temperatures. We don't know how precipitation is going to change for sure. We have models and ideas, but it's not always right, and it may get wetter in some places or drier in other places within a very short geographic range. And then the one that really shocks us year to year are extreme weather events. Excuse me. Extreme weather events are not very predictable. That's why they're extreme, right? They are more impactful where grape growers are unprepared, or any growers of any crop are unprepared for them. So, for example, spring frost events in areas where they are unexpected tend to do more damage than spring frost events in areas where they occur regularly. This can happen. I mean, if you take the example of the San Joaquin Valley, In 2022, there was a pretty bad spring frost event, which never happened at that time of year before. And it caused a lot more damage than it did in colder viticultural areas that were prepared for it with frost protection and pruning timing to help avoid that kind of damage. Prolonged heat waves are another example for the opposite situation, right? Places that are usually pretty hot are used to this and are prepared for it with their infrastructure and their practices. Cold climate areas, maybe not. They might be prepared for it because it still gets hot pretty often, but a heat wave that's above 110 Fahrenheit for 2 weeks, that might be too much for a place that doesn't commonly see extreme heat waves like that. The grapevines respond to this too, right? So the immune system of plants differs a lot from humans. Humans, we make these T cells, they stick around, they attack the pathogen. It's very good. I've liked it so far. But additive resistance is kind of what plants do, right? They make defense compounds in response to some stressor. And any stressor that is affecting them redirects the resources they need to make these compounds to deal with that stressor. So in the best conditions, grapevines can tolerate many different stressors, but there's a limit to what they can handle. And that tends to show up when there are multiple stressors. So, we just talked about abiotic stressors, frost, heat, drought, inundation. These are things that the grapevines can develop compounds for, like tannins or anthocyanins, right? Biotic stressors, they do very similar things that the grapevine has to respond to, or the grapevines respond in a similar way sometimes, right? So like animal pests, like feeding damage, plant pests, competition, or some other effect, allelopathic effects, and diseases that affect the function of the grapevine. So a vine's health and the response to stressors of any kind is a function of the available resources it has minus the biotic and abiotic stressors. And this can be seen when you look at disease expression from year to year pretty well. Host pathogen interactions are broadly impacted by environmental conditions. And because environmental conditions change from year to year, you see different symptoms year to year, right? So certain abiotic stressors, and there's also the combined stressor approach, but certain abiotic stressors can increase the susceptibility of grapevines to pathogens, or they can help trigger symptomatic expression of a pathogen. I've seen differences in expression of fungal trunk diseases in years where we've had prolonged drought and then a very large amount of winter rain the following year. It seems like sometimes that affects them. And you got to remember, and this is something that I tell, my students and I tell people I talk to, it's important to remember that we focus a lot on the grapevine's response to these things, but we should also be focusing on the pathogen and pest responses to these different conditions because they also get stressed and they also require certain resources to deal with those stressors. But the way they respond to it also affects the health and productivity of our grapevines. So the grapes are kind of in the worst situation, unfortunately. A good example of this is Xylella fastidiosa, right? The causal agent of Pearson's disease. Water stress in grapevines has been shown to increase the transmission of Xylella fastidiosa in grapevines. And this is a perfect example of a combined biotic and abiotic stressor and how plants use many different pathways and respond in many different ways to that specific combination. They tailor their response to that specific stress combination through many means, hormone signaling, receptors, transcription factors. But it might be different if the water stress wasn't drought, but flooding, and you have Xylella fastidiosa. So the response of the plant might be different in that situation, even though the two stressors are roughly the same. This is something that needs a lot more research, combined stressor responses. Bacterial adaptation, right? We're talking about how pests and diseases respond to climate change and climate conditions. Bacteria, again, they have pretty quick generational cycles. They have plenty of genetic mutations. In theory, they can adapt to new conditions relatively quickly, where a grapevine cannot. Grapevine kind of has what it has, and maybe the next generation will be able to deal with whatever stressor that the parent had, but that's not guaranteed in any way, of course. there are a lot of bacterial species in our environment already that are adapted to hot, dry conditions, and we see that in some cases. And it could affect, this is in theory, right? It could affect the localized species composition of bacteria if a competing bacteria can adapt to, say, 2 degrees hotter winters quicker than the other bacteria, which is something we should look out for, right? Just a quick note on Pierce's disease. Pierce's disease has been present in California for at least 200 years, based on records. In the 19th century, it was recorded as wiping out grapes in the majority of Southern California vineyards. And again, in the 1960s through 80s, it almost wiped out viticulture in the Temecula region again. Um, Historically, the negative impacts of Pierce's disease that we've seen were in hotter, drier, hotter, more arid regions like Southern California for those like 200 years. And that might just be because that's where we grew the most grapes, but also that is where the pathogen had the most impact, right? But we've seen these increased average temperatures. We're starting to see impacts elsewhere. There are not new, but there are newly discovered strains of Xylella fastidiosa that can handle colder winter temperatures. Um, PD has been shown to be present in a lot of places where it doesn't have a huge impact. And very often the original strain that we identified gets killed off by winter temperatures each year. And this is called overwinter recovery. Overwinter recovery from Pierce's disease or Xylella fastidiosa relies on temperatures below 53 Fahrenheit for prolonged periods, right? There's a range of temperatures and time frames where this actually helps cure it. And I don't think there's like an exact one that we can point to, but it's cold for a long time, basically. If it's warmer in winter, this could impede this phenomenon of overwinter recovery, meaning the grapevine has Xylella fastidiosa for a much longer time than it would if it was cold every winter. And temperatures in California in winter have risen by 2 degrees Fahrenheit since the 70s, which is a lot higher of an increase than the ambient temperature rise since the 70s, and made it less likely that overwinter recovery is going to occur. And then another note real quick on combined stressors. We already talked about how water stress, drought stress specifically increases transmission of Xylella fastidiosa in grapevines. These are, again, just want to bring this up one more time. These combined stressors, they create unique responses in the grapevine because of their additive defense resistance. We need to see what are going to be the most common stressors going forward in our vineyards, and how are we going to deal with them going forward? How are we going to supplement the grapevine or manage the grapevine so that it can deal with these multiple stressors that we're likely to see? Okay, so switching gears, I just want to go through a little bit of the research I've done as far as abiotic stress tolerance and climate adaptation in vineyards is concerned. And I have 3 projects that I've worked on over the past 10 years that I think are relevant for this talk and for this event. So the first is research into rootstock salinity tolerance, which I did during my PhD. Rootstock salinity tolerance is a function of the sequestration and compartmentalization of different salt compounds. This is a problem in very few places in California, but where it is a problem, it is a major problem, where the water and the soil are laden with sodium and chloride, specifically chloride. Chloride is a really big problem. So grapevines and most plants will do ion subcellular compartmentalization. It's a fancy term for saying that the cells in the roots will absorb the salt before it gets to the xylem, and they'll sequester it in their vacuoles. That's the most common method of compartmentalization that plants use. And this image here says Chen et al. 2014. This is not my paper. It just so happens that someone else named Chen with the last name Chen was working on something very similar in poplars. But it shows a really good example of where the salt is sequestered. So for poplars, you're looking at parenchyma and cortical cells, and the chlorine and the sodium get absorbed, but the potassium is allowed to go through into the xylem. So this is really interesting, which means that the plants specifically sequester these abiotic stressors that might become more common in drought scenarios where water resources are limited and the quality of water is what you have, right? You don't really have a choice in what water you irrigate with if you only have one source of water and it's limited. So we're, not expecting to, but we do see salt concentrations in irrigation water in places that have limited water resources. So this study was pretty long. I tested hundreds of grapevines to look for a suitable candidate for rootstock development for salinity tolerance. And it showed the data that I took was really focused on chlorine because chlorine is the first salt compound that causes real bad damage in grapevines and can really ruin your crop. So I focused on chlorine and I looked at the difference in chlorine uptake and deposition into root tissues and petiole tissues and leaf tissues. In the root, there was no difference, which is really interesting, right? Between all the rootstocks or wild grapevines I tested, the root tissue didn't have a difference in chlorine uptake or storage. But if we're looking at the leaf and petiole tissue, it was a very big and continuous change in the amount of sodium and chloride, but mostly chloride, that ended up in those above ground tissues, which means that these plants that are tolerant to salt stress likely exclude the chlorine ion from entering the long distance transport stream of the xylem, which is interesting, right? That is the source of salt tolerance, it seems, in a lot of grapevines. There are other sources of salt tolerance in plants, right? Mangroves are a great example. Mangroves absorb the salt in a similar fashion where they keep it in their roots, their root tissues, but they shed their roots, which is a very unique adaptation to high salinity exposure. Because the trait of chlorine uptake and chlorine exclusion in the above ground tissue was continuously variable, so that means it increases in a very steady pattern throughout all of the samples I tested, or all the different grapevines I tested, that signifies that it might be a complex trait, which means it is controlled by many different genes, not just one Punnett square on and off. It's, this, and this, then you get salinity tolerance. But if you don't have one of those, maybe you have just a little less salinity tolerance. It's very interesting. Yes, there is the continuous variation. And there is the root system where there is no variation. Fun stuff. More figures. Okay. So in grapevines, in just the rootstocks that we use day to day, right? 140 Ruggieri, 4453M, Ramsey and St. George, we looked at those as well as comparisons for the wild grapes that we were testing, so we can make sure that we don't already have salt-tolerant grapevines, right? And you can see that there are definitely delineations in the leaf petiole chlorine levels compared to the roots. Some, like 140 RU and St. George, their leaves did not sequester much of that chlorine, and the roots took most of it. And honestly, the root system didn't vary very much among all of the rootstocks I've tested, but these were the most stark examples. And you can see 140 Ruggieri and St. George were the best at excluding salt from the tissue where it would do the most damage, which is the leaves. And you can see that here too. This is the leaf and petiole chlorine accumulation when we tested it on grafted scions and on its own root system. No, grafted scions and without a scion. So this would just be rootstock in the blue and rootstock with a Cabernet scion in the red. And you can see that it's successful still given this situation. You still get lower leaf and petiole chloride accumulation in 140 Ruggieri than you do in 4453. And then a wild grapevine, Vitis acerifolia, one of the selections we had almost none. So that's likely a different mechanism. That's likely exclusion altogether, in addition to sequestration. So we do have rootstocks that are on the market today that are better or worse at salt exclusion. This is a great figure to look at that, right? And comparing blue and red, you want the red to be as low as possible and the blue to be as low as possible, but still different than the red. So what are the recommendations for salinity tolerance after I did all of these tests? For what we have out there, 140 Ruggieri, Schwartzman, St. George, and 99 Richter are the best. They're great at it if they fit your vineyard situation as far as rootability, soil adaptability, or soil compatibility, I guess, and water situation, and you have salt problems, I would use one of these. 1103P, the universal rootstock. Again, very good at salt exclusion. It's not the best, but it maintains its yield, and the fruit quality is not bad, so it doesn't accumulate too much salt. 110R also very similar. Ramsey, not great. Or Salt Creek. It took up a lot of chlorine. It had damage on the leaves, but the yield was mostly there. The worst ones are the ones at the bottom. 03916, which is a shame because we use that a lot in the North Coast. 4453, Dog Ridge, and Vinifer on its own roots, of course. Otherwise, we wouldn't be looking at a rootstock for this. So here's a snapshot of kind of my 3 years of work. Okay, the second project that I want to highlight is shade nets for sun mitigation. Another really common abiotic stressor is excess heat and excess solar radiation, which leads to berry shrivel or raisoning. I mean, in this picture, you can see half of that cluster is unusable, which means that whole cluster is unusable. But they're major concerns in California, right? Especially in places that use vertical shoot positioning and there's unexpected heat waves. Most of the time, the canopy of the grapes themselves are enough to limit damage. Which is why a California sprawl or a T-top trellis system is really good for really hot areas. But in cooler climates where you have a VSP system and you might need to do leaf removal for pest mitigation, mildew mitigation, or just sun exposure, if you get a heat wave, you get this. So leaf removal plus a heat wave means berry damage. So what do we do in that situation where we need to have sun exposure? Look at artificial shading options. Because this is what happens when you get too much sun exposure or you leave the grapes on for too long, get exposed to too much heat and sun. The real key here is that you see a really rapid drop in anthocyanins in red grapes, and you do see a drop in proanthocyanidins and other phenolic compounds too. Everything else is pretty normal on the top row, but we want to avoid this because bad anthocyanins are really bad for winemakers, especially if the consumer tends to buy by the color of the red wine, which we but we expect is true, right? So in 2016 to 2017, I looked at different colored shade nets to see which color and which transmittance pattern did best for reducing this kind of damage from heat waves and sun exposure. Overall, most of the nets, what we found was it does decrease direct solar radiation on the fruit. It decreases the temperature on the fruit. It decreases the number of sun damage clusters, and it does not reduce the quality, right? Quality of the phenolic composition or the sugar development or the acid levels of the grapes, which is great. Really, what this means is that we saw less degradation, not that we prevented any of the damage, or sorry, not that we increased the development of the fruit or improved the development of the fruit, but we decreased the amount of damage that was received during those heat wave events. And that's because it decreases temperature in direct sunlight. The shade nets were most effective during the time of day when the sun was directly on the clusters it was shading. So in the morning, the northeast side, which is the morning side for this, you can see that the temperatures were down quite a bit here by about 3 to 4 degrees Fahrenheit. During the afternoon, it actually had a pretty large impact too, where we saw a decrease of 4 to 5 degrees Fahrenheit in the canopy, which means way less damage to the fruit during the hottest part of the day, right? The red line is ambient air temperature as well. And then finally, when we looked at the visual classifications of damaged fruit, there was almost no damaged fruit when we used black shade nets with 40% transmittance. Those were the best. They performed the best. The blue shade nets with 40% transmittance were second best. The worst ones by far were the aluminum net and the white net. The aluminum net tend to look like aluminum foil. It tended to cook the grapevines. You think it would reflect the light, but it actually reflected the light back to the grapevines and cooked them. And the white shade net might as well have not have been there. It didn't do anything. So the darker shade nets with about 40% light transmittance performed best. And what we saw really was a huge decrease in the most damaged fruit. We also saw an increase in the no damaged fruit, which is fantastic. So if you have to have a VSP system and you have to have leaf removal done, artificial shading is great. It works. Make sure you pick the right shade net. Bird nets, I get this question all the time. I don't know if bird nets work or not, but these shade nets have a very specific pattern to them. It's a delta pattern, overlapping weave, and the amount of transmittance matters quite a bit too. So 40%, anywhere between like 30 and 50% seems to work fine, but 40% is what we tested, and it worked very well. I've also done light modification studies with these devices. They're made by a company called OptiHarvest. This is their OptiGrow unit. I took this LiDAR image video myself, so it's a little bad. But this is more or less what they look like. I just wanted to give you a 360 picture. And these go on young grapevines, right? Things that aren't trained to the trellis yet, things that are just vertical, like a trunk and maybe a head of a grapevine. And they're really good at scattering and diffusing light while limiting direct solar radiation. The real kind of important thing here is their shift in temperature and their shift in the light regime. Again, this is to limit not only damage from heat and sun, but also to increase temperatures in the canopy during the development of a young grapevine, right? I see these as being very useful for replants. Filling a whole vineyard with them might be a challenge, but if you have to replant 2% of your vineyard, these would be a great option to get those vines to production a little faster. What we did see was on the hottest part of the day, so this figure on the right here, these are 3 days over August, and the last 2 days here were heat wave days, so above 100 degrees Fahrenheit. And you can see that the hottest part of the day, the control with no cone on it, got really hot in the canopy. The treated units, which are the blue and the red here, they reduce temperatures by 3 to 5 degrees Fahrenheit, which can make a huge difference during a heat wave, especially if there are water limitations. I also looked at the light regime in these because photosynthesis requires, a range of light, 380 to 720 or 700 about nanometers. And I did this by looking at the light intensity spectrum in the open field versus a control grapevine, which is this, right? So this is underneath the first leaf layer of the grapevine. This is how much light a single leaf layer in a vineyard absorbs. Most of it is within that 380 to 700 nanometers, which are the visible light spectrum. And you can see a spike around the green area, which is what they reflect. And that makes sense, right? Infrared light is also reflected quite a bit. With the OptiGrow units, a lot more light was absorbed. And my take on this is that there was less direct exposure of sunlight because it's surrounded by this cone, right? But the cone is designed to redirect or diffuse light around the canopy, which means that you get more of this red, orangish red light in the canopy, which seemed to have a lot of good benefits. So one of those was increase in shoot length and shoot diameter and a decrease in water stress, which was awesome. So by the end of the season, during the season, the differences between the control and the cone were minimal as far as shoot length and shoot diameter go, but by the end of the season, they were significant, which was really cool. The 2%, which is 2% pigment level, OptiGrow unit increased by about 10 to 15% in shoot length and about 10% in shoot diameter, which is really good if you're trying to retrain a vine quick in a cold region, right? It also protects against heat waves. And then water stress was also different, but it was mostly just lower slightly in the units. So that was nice to see. The vines are just a little happier there than they are on their own in the field. So these are useful for replants, it seems. They make other products I haven't tested, but they all use the same kind of technology. They help decrease temperatures during heat waves, which is great for climate adaptation. They help increase temperatures during cold periods, which is also good for cold snaps in spring and fall. They can increase the shoot growth as far as length and diameter go of 1 to 2 year old vines. They decrease water stress on average throughout the year. And likely they're increasing the total light availability in the entire vine, right? So they reduce direct light, increase diffuse light, and that goes back to this image here. It's bouncing light around throughout the canopy, which is really interesting. That way you don't get just that first leaf layer getting everything. You get a little bit of some light exposure throughout the entire young grapevine canopy. So this seems to mitigate the impacts of extreme weather events and can be really useful. The last study I just want to briefly go over is a 2.5, 3 year study I was looking at regarding rootstock selection for drought recovery. And originally, this project was designed as a rootstock selections for drought tolerance. But when I started it, we got a lot of rain. So it became for drought recovery because before 2022, we had several years of, pretty dry conditions during winter. I did this across 9 vineyards in California's North Coast region, across 3 counties, Sonoma, Mendocino, and Lake County, equally distributed among them, and looked at Cabernet Sauvignon and Chardonnay as options for scions and 5 different rootstocks. The goal was to see which rootstocks could recover best from prolonged drought periods in the next year or two after they get enough sufficient water during the rainy season. And then the other main objective was to identify site characteristics that might impact a grapevine's recovery from drought conditions. Right? In California, we get these cycles of drought and good precipitation. I wanted to see which grapevines perform best in those conditions. Right? Water stress was a gimme. It was very important to measure how stressed the vines were. And this is just one scion. I believe this was Cabernet, and how it performed with different rootstocks. And you can see that these were recoveries of the water potential or decrease of stress after the last irrigation event. That's what I wanted to capture here, as well as throughout the year how it performed. With the last irrigation event, 110R recovered the quickest, and that makes sense because it has both a shallow and a deep root system. It can take up water quick and it can get water deeper. 3309 and SO4 were the slowest. Again, shallow root systems, if they don't have a lot of water, they don't get a lot of water. And the water should be applied slowly for shallow root system rootstocks. But during water stress times of water limitations, SO4 seem to be the most stressed. I looked at cluster counts and yields too, because the development of clusters really depends on the conditions for the year before. There wasn't much of a reliable difference here, so I'm going to not talk about that too much. The vineyard geography mattered quite a bit. The coastal valley regions were delayed in their development every year for the past 10 years or so. And the inland mountainous and inland valley regions, they developed around the same time every year. So the conditions varied from site to site. So I was like, I have this device, let's measure the light intensity at each of these places. I got that device specifically for the OptiGrow project, but I was like, okay, let's do it here. You can see the difference in light spectrum intensity in the different geographic classifications I had. So the inland valleys, they got more of that bluish, greenish light, and the coastal mountains and inland mountains got a lot more red, infrared light, which changes how grapes grow, right? The grapevines need a lot of red light to grow vegetative tissue. I also looked at the site water availability, and the key takeaway here, as far as left is evapotranspiration, right is precipitation. The key takeaway here is that the inland mountainous regions had higher water loss through evapotranspiration, and every year they had lower precipitation. So inland mountain regions have much less natural water falling on them, and they lose a lot more water than the other 2 region types, coastal valley and inland valley. And you can see that here, this area and this area, as far as the growth rate and the evapotranspiration. So overall, just some quick takeaways from the research. For heat stress and solar radiation stress, artificial shading is great. It is very useful if you have a limited way to grow the grapes and you can't change your trellis system or your leafing practices. You can also use light modification technology to limit the impacts of these extreme weather events like cold snaps or heat waves, or just poor light availability if you grow grapes in a cloudy or foggy region. And for abiotic stress tolerance, rootstocks are very important. They were significant. The choice of rootstock was significant in every physiological metric that I measured during my rootstock drought recovery study. But the site conditions were the most important, and the geographic classifications were most important for abiotic stress tolerance as it relates to water. So, it's the old saying, location, location, location. So my last few slides are how do we utilize rootstocks for climate adaptation? So rootstock and scion selection is really important to adapt to the different conditions and preserve our viticulture areas. We need to look at different varieties that might perform better there as the conditions change. I always recommend trying to plant experimental vines. If you have the land for it, you have the resources, plant a couple of random varieties that you think might do well as climates change and based on what it's expected to change as or change toward. And then by the time you're ready to replant, you can see which one performed best. And if you have a market for it, by that point, you're in the clear. Sticking to the same rootstocks and the same scions can be problematic going forward. This is an example of that. The different root systems of a deep rooting rootstock and a shallow rooting rootstock with very different tolerances to drought. If your site, your location is becoming drier over time, you might want to switch from like a 10114 to a 140 RU or a 110R. It's important to make a note of saying that less is not more. As far as the scion varieties go, it's very understandable that we have such a dominance of Chardonnay and Cabernet Sauvignon. But rootstocks, we have a lot more wiggle room, and I think we should be exploring those quite a bit. And we can do that by looking at where, scions were developed, what rootstocks were developed for. Do they come from hot regions? Do they come from cold regions or from moderate regions? How are the conditions where they were developed? And does that match up with your conditions now and the expected conditions at your vineyard in 10, 20 years? We can modify our trellis design. VSP is very useful and very necessary in some places, but before VSP took over, California sprawl and T-tops were the most dominant trellis designs in California because they shade the fruit themselves. Can we go back to that? Maybe. It depends on the situation, your climate, and whether or not you have the machines or the labor to do it. We need to monitor pests and diseases, right? LiDAR identification is new. There's drone-based aerial imagery, and there's ways to improve your site monitoring that are less high-tech than that. I think monitoring, that is the takeaway from how we manage new pests and existing pests going forward. And then here are some climate adaptive approaches. I want to leave some time for questions, at least a minute or two. So I'm going to leave these here. And I'm going to say you can find this presentation at this website under speaker presentations on the left bar. That is where I upload all my presentations. So there's more there than just this. And just a quick thank you to everybody who's helped me with these projects, Andy Walker, Megan Bartlett, all the great growers that helped me in the region, and all of the foundations that helped fund this research are here. So thank you very much. Happy to take some questions.
Thank you, Chris. We have 2 minutes for questions. First one, did I hear you say that lower winter chill hours can affect bud break? I thought all grapes were very low chill, less than 200 hours.
Yeah, it's uncommon in California especially, but there are regions of California where chilling hours are so limited in winter that there is a requirement to use chemical intervention to induce bud break in spring. It's not very common. It's kind of more of a watch out for this thing, but if you're in the North Coast, that's probably not something you have to worry about going forward. It's those really hot regions that don't get very cold in winter that worry about that. Yeah, it's quiescence is the term. Quiescence is a big requirement of grape buds to break, and it does have a very low bar. So not something to stress about, definitely.
Could you please go back to the presentation slide? I'm not sure.
The first one?
I'm not sure. It's not very clear. But the next question is, for rootstocks that perform poorly with salinity, could you please describe the root development?
Yeah, so rootstocks that are bad with salinity, the roots tend to grow fine for a while. It's not a matter of deep rooting or shallow rooting, although in a vineyard, a deep rooting rootstock tends to be better for salinity tolerance because water deeper down tends to not be as salty as what we apply to the surface. The issue with grapes that are not salinity tolerant is that they don't hold on to the salt in their roots. And the ones that do hold on to salt and are salinity tolerant, they can end up losing some of their roots to cell death. So you end up with this three pathways you can go down with root systems and salinity tolerance. You can have the ones that don't hold on to anything, they grow fine, and then the above ground tissue dies. You can have the ones that hold on to a lot of salt and then the roots die, which is also bad. Or you can have the ones that do a good in between where they exclude some salt, they hold on to some salt, the roots don't die as often, and the above ground tissue doesn't get salt accumulation as much. That third one is what we're going for. It's not so much like the architecture, it's more of how what mechanisms they have to respond to temperature, I mean, to salt.
So they're asking to go back to the slide that shows the URL for your presentations where they can find
Oh, okay.
questions. If you have one more we're at time. You have one more question that's in the Q&A. If you don't mind jumping in and answering that by
Sure.
typing in. Thank you, Chris.